Hi, we're continuing with my notes for life science cell biology in a nutshell. We are learning about what goes on in, well, mostly the nucleus, where you have DNA, which is genetic material, and the DNA encodes for certain, it encodes for RNA, and the RNA encodes for proteins. This is used for making of proteins important for the cells, as well as in the replication cycle. We were looking at one example of DNA encoding for a specific type of protein molecule. This is the antibody molecule, and different kinds of DNA can encode for different kinds of such molecules. So we have the subunits of this protein. So it says the light chain and the heavy chain in blue and pink, respectively, is encoded by different parts or sections of the DNA. So, okay, and this is just a major protein type that is encoded by DNA that is important, and sometimes variation of this molecule is helpful. Um, Over time, selectivity for more favorable variations of this molecule can help, um, uh, bind different kinds of antigens which need to be bound um, in the immune system. So different kinds of proteins need to be coded in response to different kinds of antigens. Um, so the DNA segments undergo considerable rearrangements, resulting in the synthesis of a great variety of antibodies, which are proteins. Um, so these, the binding sites on each molecule of antibody is formed by one light and one heavy amino acid chain, which are encoded by different segments of the DNA in the lymphocyte nucleus. So the immune system means that given a time, the host organism can overcome an infection by producing antibodies to the parasite's glycoprotein coat. So whatever protein is on the outside of on the coat of the membrane, these antibody can bind that protein, and uh, clear your body of that protein that you don't want. Comparisons of gene structure have revealed epigenetic modifications, heritable changes that occur on the sugar phosphate side of bases in the DNA and thus do not cause rearrangements in the DNA sequence itself. An example of epigenetic modification involves addition of a methyl group to cytosine bases. This appears to cause an inactivation of genes that do not need to be expressed in a particular type of cell. So, there's ways of controlling how the DNA is expressed, depending on what kind of cell it is, of what kind of tissue it is in, what the needs of that cell are, and at different parts of the lifespan of a cell, what it's undergoing. Different proteins need to be made. Therefore, different parts of the the DNA needs to be transcribed into mRNA than proteins. So, are expressed. So, cytoskeleton, of course, was what we learned about earlier, um, and this is what keeps the cell intact, and controls its interior parts. It controls um, the cells changing of shape, and during replication, especially, it helps in the shuttling of different molecules from across the cell, from different from one location to another, via proteins and myosin protein motors, etc. There's nice pictures showing the microtubule here, which um, composed of protein tubulin component of the cytoskeleton. This is involved in the intracellular transport or cellular mobility and nuclear division. As I said, very beautiful picture of the cells, cytoskeleton, protein fibers. So intracellular, meaning inside the cell. This is showing a 3D of the microtubule molecule, which is made out of beta tubulin and alpha tubulin, comprising a dimer. That's what it looks like. It's like a long cylindrical filament. These are shown in, these are the cells. I believe in the biggest part of the cell, which is in blue, is the interior, and microtubules are in green. They play a role in cytoplasmic streaming, which is movement of the cytoplasm, which contains all the organelles of the cell, as well as molecules, proteins, and other kinds of molecules involved in the different processes of the cell. Dystinin is a protein which anchors neutral intermediate filaments to actin cytoskeleton. So these are 3D renderings of the molecules, just to get an idea of how complex and how large they are in space. As that's what the molecules look like when they're all in their 3D conformations. Dystinin is a protein anchoring. Okay, that's dystinin. This is dystroglycan, a protein which works as a transmembrane linkage between extracellular matrix and the cytoskeleton. Transmembrane being it links the outside of the cell to the inside of the cell. So that's a very important. So that in making of tissues, so linking of cells, and bringing the inside and the out, linking the inside and outside of the cell together. Various reasons, maybe shuttling molecules or whatnot. Spectrin is a molecular scaffold protein linking plasma membrane to actin cytoskeleton. Functions in determining of cell shape and arrangement of the transmembrane proteins. So transmembrane is whatever proteins are inside the membrane of the cell, the outer double membrane of the cell. Ankyrin R is membrane of ankyrin, some family of adapter proteins mediating attachment of integral membrane proteins to the spectrin, actin-based membrane cytoskeleton. So again, the cytoskeleton, which is the skeleton of this inside the cell, basically made of the protein fibers linked to the membrane itself. We'll continue